Have you ever wondered why some diseases are passed down through generations? We're talking about genetic disorders, conditions rooted deep within our DNA. These disorders can greatly impact human health, often affecting individuals from birth. Genes, the building blocks of life, play a crucial role in determining our health and well-being. At times, alterations in these genes can lead to severe health conditions. It may seem like genetic disorders are an unchangeable life sentence, but science is constantly evolving and working on solutions. Genetic disorders may seem like a life sentence, but science is working on solutions. To understand how we can treat genetic disorders, we first need to know what they are. Genetic disorders are diseases caused by abnormalities in an individual's genetic material or genome. Our genes, which are the building blocks of our DNA, determine our physical characteristics and how our bodies work. So, when there's a mutation or alteration in these genes, it can lead to a variety of health problems. Let's consider 3 examples of such disorders: cystic fibrosis, Duchenne's muscular dystrophy, and hemoglobinopathies. Firstly, cystic fibrosis. This is a disorder caused by a single gene mutation that affects the CFTR, or cystic fibrosis transmembrane conductance regulator. This mutation disrupts the normal function of the lungs and digestive system, leading to a buildup of thick, sticky mucus, which can cause severe respiratory and digestive problems. Next, we have Duchenne's muscular dystrophy, another single gene disorder. This time, the mutation occurs in the dystrophin gene, which is crucial for muscle function. When this gene is mutated, it leads to progressive muscle weakness and loss, affecting both skeletal and heart muscles. Lastly, hemoglobinopathies. These are a group of disorders, like sickle cell disease and thalassemia, caused by mutations in the genes that code for hemoglobin, the protein in our red blood cells that carries oxygen throughout our bodies. These mutations can lead to a variety of problems, from abnormal red blood cell shapes to reduced oxygen-carrying capacity, causing severe anemia and other complications. In each of these cases, the root cause is a single gene mutation. It's like having a blueprint for a house, but somewhere along the line, a mistake was made in the plan. This mistake then translates into the final structure, causing it to function less efficiently, or not at all. With this understanding, we can now explore how science is fighting back. Enter CRISPR/Cas9, a revolutionary tool in genetic engineering. This extraordinary technology has the potential to correct genetic mutations, potentially transforming the way we treat diseases like cystic fibrosis and Duchenne's muscular dystrophy. Let's dive a little deeper. CRISPR/Cas9 is like an ultra-precise genetic surgeon, able to target and modify specific genes within our DNA. This technology opens up the possibility of correcting genetic disorders caused by single gene mutations. A study led by Schwank and his team turned to CRISPR/Cas9 to explore the treatment of cystic fibrosis. They started with adult intestinal stem cells from 2 cystic fibrosis patients, and using CRISPR/Cas9, they managed to correct the mutation that causes the disease. The outcome? The restored function of the CF transmembrane conductor receptor, a crucial protein that malfunctions in cystic fibrosis patients. Another disease, Duchenne's muscular dystrophy, has also been under the CRISPR/Cas9 microscope. Tabor Bordbar and his team used a delivery vehicle known as adeno-associated virus to introduce CRISPR/Cas9 into a mouse model of Duchenne's. The result was a partial recovery of muscle function, thanks to the production of a truncated but still functional dystrophin protein. This was a significant finding, especially since the dystrophin gene was edited in muscle stem cells, which replenish mature muscle tissue. This could mean that the therapeutic effects of CRISPR/Cas9 won't fade over time. What's more, there are other studies that have successfully used CRISPR/Cas9 to increase the expression of the dystrophin gene and improve muscle function in mouse models of Duchenne's. Some research even suggests that this approach could lead to the production of full-length dystrophin in human patients. And it doesn't stop there. CRISPR/Cas9 could also be a game changer in treating hemoglobinopathies. By disrupting a specific gene enhancer, researchers have managed to induce fetal hemoglobin in mice and human cells, paving the way for a novel therapeutic strategy in patients with diseases such as sickle cell disease or thalassemias. CRISPR/Cas9 is not just a scientific breakthrough, but a beacon of hope for those with genetic disorders. The future of genetic disorder treatments looks bright thanks to this remarkable tool. While CRISPR/Cas9 holds great promise, there are other potential treatments on the horizon. One such treatment is BCL11A enhancer disruption. This approach may hold the key to treating hemoglobinopathies, disorders that affect the protein in red blood cells that carries oxygen. Recently, a study demonstrated that BCL11A enhancer disruption, achieved using CRISPR/Cas9, could induce fetal hemoglobin in both mice and human erythroblast cells. Fetal hemoglobin, unlike its adult counterpart, is not affected by certain genetic disorders. This means that, in the future, we could potentially express fetal hemoglobin in patients with abnormal adult hemoglobin, offering a novel therapeutic strategy for diseases like sickle cell disease or thalassemias. The process of knocking in a fully functional B globin gene, however, is more challenging. This is why researchers are exploring this somewhat unusual approach of using fetal hemoglobin. It's important to note that these treatments are still in the experimental stage, but they represent a significant step forward in the fight against genetic disorders. The success of these experimental treatments hinges on ongoing research and development. The future is shaping up to be an exciting one as we continue to unlock the secrets of our genes and learn how to correct the errors that can lead to disease. The future of genetic disorder treatments is bright, filled with promise and potential. In the past 7 minutes, we've journeyed through the complex world of genetic disorders. We've explored the intricate science of these conditions, revealing how singular gene mutations can lead to diseases like cystic fibrosis, Duchenne's muscular dystrophy, and hemoglobinopathies. We've delved into the revolutionary technology of CRISPR/Cas9, a tool with the potential to rewrite the story of genetic disorders. We've seen its potential in correcting mutations, restoring protein function, and even inducing fetal hemoglobin in both mice and human cells. The future of genetic disorder treatments is full of promise. With CRISPR/Cas9, we're looking at a new era where genetic conditions could be treated at their source. It's a future where Duchenne's muscular dystrophy patients could have a functional protein, where cystic fibrosis patients could have a restored CFTR function, and where hemoglobinopathy patients could express healthy hemoglobin. Science is tirelessly working to turn the tide on genetic disorders, and every day we're getting closer to that goal.